In the name of God, hi to everyone. I'm Dr. Majid Mujarrad, Associate Professor of Medical Genetics from Mashhad University of Medical Science. It's my honor to give you a speech about CRISPR-Cas system. We will discuss about basics of CRISPR-Cas and new achievement in this era. The age of recombinant DNA technology or genetic engineering was started from 1973 when Professor Herbert Boyer and Stanley Cohen can introduced Xenopus levis DNA into bacterial genome. As a brief, in genetic engineering, we use a gene of interest from a cell and introduce that into the recombinant DNA, such as plasmid, and transfer the recombinant DNA or recombinant plasmid into the host cell. This host cell can be a bacterium or a eukaryotic cell. Then we produce a product. This product can be a recombinant cell or a recombinant protein, such as human growth hormone as a recombinant protein and the streptokinase as a recombinant protein, or insect resistance plants, or a bacteria that can clean up toxic waste from water resources. We can call this progress as a genetic engineering version 1, but this version has some major limitations such as size of DNA. Theoretically, we can manipulate 150 kb of DNA as a maximum, but technically we can efficiently engineer up to 20 kb of DNA fragments. We must manipulate DNA as an episomal DNA and not in genome level. Furthermore, there is a risk of random integration for foreign DNA. Main reason of this limitation is the use of restriction enzyme. Restriction enzyme has a recognition site of 6 to 8 base pair. This leads to cutting of genome around every 4 to 50 to 65 kb. So genome manipulation is not feasible for complicated genomes such as human genome. We need a restriction enzyme with a 18 base pair recognition site for manipulating of such a large. So genetic engineering must enter to version 2. This version was based on genome editing using homologous recombination. Using homologous recombination, we can introduce modifications into endogenous genes. That leads to disruption, insertion, or the replacement of a locus of genome, which can control gene expression, create SMP, and create reporter fusion while maintaining endogenous gene regulation. However, homologous recombination is not an easy choice. This process isn't common in normal cells, so it can be done in a few types of cells, and this make these experiments time-consuming with ultimately low efficiency. To increase of homologous recombination, we must induce a double-strand breakage in desired site. But finding of an enzyme that makes this unique cut in genomes is a hard target to achieve. 1 decade after using of homologous recombination for gene manipulation, zinc finger nuclease or ZFNs were discovered and were used to targeting DNS. And 1 decade later, talents were introduced to doing this mission. These artificial restriction enzymes have two parts, DNA recognition part and cleavage domain, which come from FOC1 nuclease. As used in zinc finger nuclease, two zinc finger nuclease complexes will bind to DNA strand on opponent strand, and the dimerization of FOC1 subunit leads to double strand. Talons also work similar to ZFNs. However, presence of repeated modules which recognize each base pair of DNA make designing of talons easier. FOC1 induced double-strand breakage may lead to either of non-homologous end joining or homologous recombination. Non-homologous end joining is more common and usually cause insertion deletion mutation, which eventually leads to homology-directed repair or homologous recombination occur when a donor sequence is present. This process leads to precise manipulation of DNA, such as targeted gene integration or site-specific mutagenesis. While huge progress in genome manipulation using these methods, ZFN and talon, but protein nature of these tools make them very expensive and time-consuming. Furthermore, failure rate is very high and they show very high off-target rate. Many efforts were focused on the finding of a tool who have high specificity and also easy to make. And this dream became a reality in 2013 with introducing of CRISPR-Cas system. CRISPR sequence were discovered in 1987 from E. coli, and until 2000, similar repeat of CRISPR were identified in other bacteria. In 2002, the sequence and the associated protein were named CRISPR and Cas, and in 2005, it was clear that CRISPR elements are derived from bacteriophage. In 2007, it was discovered that CRISPR sequence can alter resistance of Streptococcus thermophilus to phage attack, and it was clear that these sequences are a part of adaptive immunity of bacteria. Finally, in 2030, two independent research teams, George Church and Feng Zhang from Broad Institute of MIT, and Emmanuel Carpenter and Jennifer Doudna from Berkeley University, almost simultaneously introduced CRISPR as a breakthrough discovery to editing of genomes. So, what is CRISPR-Cas? These, in fact, are adaptive immunity of bacteria and archaea against phage. CRISPR is the abbreviation of clustered regulatory interspersed short palindromic repeat, and Cas protein is abbreviation of CRISPR-associated proteins. These are part of adaptive immunity that evolved to adapt and defend against foreign genetic material such as phage in bacteria and archaea from different type of bacteria. Several different of CRISPR pathways were discovered, but type 2 of CRISPR named CRISPR-Cas9 creates a double-strand breakage in the target of CRISPR has four basic parts: protospacer adjacent motif or PAM, which is a 3 or more nucleotide downstream of recognition site of sgRNA. CRISPR RNA or cRNA which recognize a sequence of upstream of PAM, and tracer RNA, which bind to cRNA and direct Cas to site of recognition, and Cas protein, which binds to this site and cuts the CRISPR-Cas system produce and act in three stages. In stage 1, all CRISPR spacer acquisition, Cas1 enzyme cuts foreign DNA into short fragments and integrate that to new spacer sites in CRISPR locus on host gene. In stage 2, CRISPR is expressed from genome as a precursor cRNA and then cut to individual cRNA. In stage 3, cRNA hybridizes with tracer RNA via its complementary segment and produce an RNA complex. Then this RNA complex bind to Cas protein, and this RNP complex recognize phage target site and cut it out. Binding of tracer RNA, cRNA, and Cas to a specific site of genome leads to double-strand breakage of targeted site of this genome. FOC1 induced double-strand breakage may lead to either of non-homologous end joining or homologous recombination. Non-homologous end joining is more common and usually cause insertion deletion mutation, which eventually leads in stage 2, CRISPR is expressed from genome as a precursor cRNA and then cut to individual cRNA. This capacity of CRISPR make CRISPR-Cas strategy an ideal tool to gene therapy. In less than 2 years of introducing CRISPR, several transgenic animals were produced using this strategy, from Drosophila melanogaster to transgenic monkey and even human embryo. Furthermore, CRISPR-Cas strategy was successful in treating of Mendelian disorders, such as cataract in a mouse model, and also treating of CF in human intestinal stem cell organoids of CF. As an inspiratory achievement in 2014, CRISPR succeeded to cure tyrosinemia in a mouse model. This achievement was very exciting because it was performed in vivo. In next years, CRISPR system was entered in post-genome era. CRISPR was used in epigenetic manipulation of genome, gene suppression, gene activation, and also conditional gene expression regulation. Main reason of this limitation is the use of restriction enzyme. Restriction enzyme has a recognition site of 6 to 8 base pair. This leads to cutting of genome around every 4 to 50 to 65 kb, so genome manipulation is not feasible. Recently, in 2017, engineered Cas proteins open a horizon to precision base editing, which can correct a single base mutation. This can be a tool to treat genetic disorder by correcting of point CF in human intestinal stem cell organoids of CF. By use of CRISPR systems and may other new gene editing strategies, as Jennifer Doudna says, we may be nearing the beginning of the end of genetic disorders. Thank you.